This tutorial is going to walk you through how to approach this question. If you haven't read it over yet, please pause the video now and take a minute to read the question, then come back to see how we're going to approach it. Part A of the question is asking us to determine if each vitamin would be soluble in the aqueous environment of the cell. In other words, is each molecule predominantly hydrophilic, water-loving, or hydrophobic, water-fearing? How can you tell? When we're looking at molecules in biological systems, we answer this question by determining if the molecule contains the kinds of functional groups that would allow it to interact favorably with water. Most functional groups in biological molecules contain oxygen or nitrogen, and so have polar bonds which make them hydrophilic. In contrast, carbon-carbon and carbon-hydrogen bonds are nonpolar and do not interact favorably with water. Molecules that contain only carbons and hydrogens, or mostly carbons and hydrogens, are generally hydrophobic, while those that contain oxygen and nitrogen can be hydrophilic. So back to our question, let's start with vitamin D. What we see here is a simplified line drawing representing this molecule. Remember that in this type of depiction, straight lines represent bonds, and the ends and intersections of any lines that are not otherwise labeled indicate the positions of carbons, with enough hydrogens present to satisfy the octet rule. In other words, anytime no other atom is indicated, we can assume that it's a carbon-carbon or carbon-hydrogen bond. So at this end, we would have a CH3 group. This intersection is CH, and this would be CH2. The rest of this molecule is more carbons and hydrogens. Since both carbon-carbon and carbon-hydrogen bonds are considered nonpolar in biological systems, this molecule is almost entirely nonpolar. In fact, the only functional group is all the way down here. This single polar group is insufficient to give significant polar character to this molecule, so it is considered hydrophobic, not soluble in the aqueous environment of a cell. Let's contrast that with vitamin C, shown here. These red dots indicate carbons that are participating in polar bonds because they are attached to oxygens, which are electronegative enough to make polar bonds with carbon and hydrogen. There are many polar functional groups in this molecule, so it is hydrophilic. It would be very soluble in the aqueous environment of a cell. Vitamin B6 is a bit more of a complicated situation. Some of the carbons are attached to functional groups that are polar and that form polar bonds, but others only form carbon-carbon and carbon-hydrogen bonds, and these carbon-nitrogen bonds here, because they are part of this ring, are not especially polar. Nonetheless, there are a number of polar functional groups found attached to various carbons, and no particularly large portions of the molecule lack functional groups, so this molecule would be classified as hydrophilic. Now let's look at part B. A key definition that you need to know to answer this question is what it means for something to be deprotonated or protonated. This means that the molecule or functional group either has a proton, a hydrogen ion, H+, or it doesn't. We usually use these terms to refer to acids and bases. Acids can lose protons or become deprotonated, while bases can pick up protons and become protonated. If we look at our example acid here, vitamin C, the neutral form, the one that still has the proton, is considered protonated, while the ionized form, which has given up its proton and so has a negative charge, is deprotonated. In contrast, if we look at a basic group like an amino group, the neutral form is deprotonated, in this case because it lacks an extra proton. The protonated form has picked up an extra H+, and so has a positive charge. This is a key distinction to remember. Ionized acids are deprotonated and have negative charges, while ionized bases are protonated and have positive charges. So now that we have that sorted out, we need to think about what's going on with this particular vitamin C molecule at different pHs. Recall that pH is a measure of how many protons are in a solution, and pH can be acidic, basic, or neutral. When we think about solutions, those that are acidic have excess protons. Those that are basic have lower concentrations of protons. And based on the rules of chemical equilibrium, a molecule would tend to be protonated in solutions with high proton concentrations and deprotonated in solutions with low proton concentrations. Since pH is a measure of the negative log of the proton concentration, lower pHs, or those that are less than 7, are found in acidic solutions, which have excess protons and in which molecules will tend to be protonated. Higher pHs, or pHs greater than 7, are found in basic solutions, which have fewer protons and in which molecules will tend to be deprotonated. There are two specific pHs referred to in the question. pH 3 describes an acidic solution, one with excess protons. Because of all those extra protons, the molecule is more likely to be protonated at this pH. pH 10 describes a basic solution, one with fewer protons. Because this solution is proton deficient, the molecule is more likely to lose its proton or to be deprotonated. And now we can move on to part C, which asks about functional groups. For this class, there are a relatively small number of functional groups that you're expected to know. Hydroxyl groups are very important in biology, so you should be familiar with them. Carbonyl groups as well. If you add an OH group to the carbon in a carbonyl, you get a carboxyl group. These are common in biological acids because this proton here will often be lost and the molecule deprotonated at physiological pH. Then we have amino, phosphate, and sulfhydryl groups. Amino groups, as we saw earlier, can pick up an extra proton and gain a positive charge, while phosphate groups tend to lose these protons, gaining a charge of -2 overall. Sulfhydryl groups are actually a bit unique among these functional groups in that they are not particularly polar. They will not make a molecule hydrophilic, for example, but they do have other important characteristics that we'll talk about more when we get to proteins. Once you know these functional groups, they should be pretty easy to identify in biological molecules. In vitamin B6, we have 3 of them. This hydroxyl down here, the carbonyl here, and the phosphate group here. And that's how you can work your way through questions that ask about interactions with water, the behavior of molecules at different pHs, and different functional groups. If you have any questions, you can post them to the discussion forum for Lecture 1 or come to visit us in office hours to talk about them.